Imagine a hacker who doesn't target your bank account or your computer's operating system, but targets your very own DNA. We have long defined national security by physical parameters, sovereign borders, airspace defense, and standing armies. But as we settle into the biological century, the primary theater of conflict is shifting from the external world to the microscopic domain within us. We are moving rapidly toward a reality of hacked borders, where biological code is as programmable as software, and the concept of a border is transforming into a dynamic genetic perimeter. The catalyst for this shift is a powerful, largely invisible convergence of artificial intelligence and synthetic biology. In early 2025, the landscape changed with the release of open-source AI models like Evo2. These models were trained on an unfathomable 9.3 trillion nucleotides from over 100,000 species. This computational power allows for simultaneous reasoning over protein sequence structure and function. Effectively, we have turned biological research from a process of discovery into a search and edit operation. Historically, developing a sophisticated biological agent required the massive resources of a state-sponsored program, high containment labs, and years of trial and error. Today, generative protein design tools allow researchers to create entirely novel protein sequences that have never existed in nature. This is the black box of modern biology. Traditional biosecurity relies on homology-based screening, essentially checking a database to see if a sequence looks like a known toxin, like anthrax or ricin. But AI can now design proteins that perform hazardous functions while looking nothing like any known pathogen. These are next-generation agents, and they are currently invisible to our standard security scanners. This brings us to the dual-use paradox of CRISPR technology. On one hand, CRISPR is the medical miracle of our time, offering the potential to cure hereditary diseases and cancer. On the other hand, it has democratized the ability to manipulate life itself. CRISPR kits are now available for purchase online for less than $500. We are seeing the rise of a global, it-yourself biology movement, where enthusiasts experiment with gene editing in community labs or even private garages. While this open innovation drives science forward, it bypasses the oversight structures established by the 1972 Biological Weapons Convention. That treaty was written for a world of nation-states, not for a world where a graduate student or a rogue actor could potentially engineer a pathogen in a basement. The threats we face in this new era are distinct from the biological weapons of the Cold War. We are looking at agents that are small in scale but sensational in nature. These engineered pathogens can be modified to induce stealthy latency, lying dormant in a host for weeks or months before triggering symptoms. This capability creates a fog of engagement. If an outbreak occurs, it becomes nearly impossible to distinguish between a natural mutation, a laboratory accident, or a deliberate attack. This ambiguity erodes the very concept of deterrence. You cannot retaliate against an enemy if you cannot prove they fired the shot. Even more disturbing is the theoretical potential for ethnic bioweapons. While scientifically difficult, the data suggests that as we map the subtle genetic variances between populations, it becomes theoretically possible to design agents that preferentially target specific genotypes. Historical precedents, such as the darker chapters of Project Coast in South Africa, show that the intent to create such weapons has existed before. Today, the technology to execute that intent is closer than ever. So, how do we defend a border that exists inside our cells? The strategy is shifting from physical interdiction to safety by design. The U.S. Defense Advanced Research Projects Agency, or DARPA, has launched the Safe Genus program. This initiative is not just about editing genes, but about controlling the editors. They are developing genetic off switches and reversible inhibitors that can neutralize a gene editing tool inside a living system. Imagine a biological fire extinguisher that can stop a gene drive or a CRISPR attack in its tracks. Simultaneously, we are seeing the deployment of pathogen-agnostic metagenomic sequencing. This is a surveillance dragnet for the air. Unlike a COVID test that looks for one specific virus, this technology sequences everything in a sample, from airport wastewater to the air filters on a plane. It uses high-performance computing to flag anything that looks genetically anomalous. Programs like the CDC's Traveler-based genomic surveillance are already live in major international airports, acting as a radar system for biological threats entering the country. However, this total biosurveillance comes with a profound price tag: our privacy. To catch a thief in the genetic code, the government must scan the code of the innocent. We are witnessing what legal scholars call raiding the genome. Reports indicate that border agencies have collected DNA samples from hundreds of thousands of individuals, including asylum seekers and citizens, who have not been charged with crimes. These profiles are being amassed in vast federal databases, like CODIS in the US or the massive genetic registries in China, which reportedly hold data on 80 million citizens. We are entering a Gattaca-like reality where your biological data is the ultimate identifier. If the government scans wastewater or air filters to find a virus, they are also inadvertently capturing the human DNA shed by everyone in that space. This creates a bypass around the Fourth Amendment and other privacy laws. We are rapidly building an infrastructure of surveillance that is nearly impossible to opt out of. We stand at a crossroads. The same bio-hacked world that promises to end genetic disease and secure our food supply against pests also threatens to turn our own biology into a battlefield. The treaty gap is widening, technology is moving instantly while diplomacy moves incrementally. The question for the next decade is not just about whether we can survive the next pandemic, but whether we can survive the measures taken to prevent it. Are we willing to trade the privacy of our own genetic code for the security of the herd? I want to know where you draw the line. Does the threat of engineered viruses justify total genetic surveillance? Let's debate this in the comments below. If you want to understand more about the technologies rewriting the rules of life and war, make sure to like this video and subscribe. The future is being written in A, C, T, and G, and we all need to learn how to read.